Okay, and I'm just going to give you some information to help you complete the lab and understand what's going on. So let's say that this is inside the nucleus of the cell. We have had the chromatin condense into chromosomes, and we're preparing for meiosis. So here's one chromosome, here's another chromosome. Now in a diploid organism, diploid meaning that you have two sets of chromosomes, you have one set from one parent and one set from another parent. In each of those sets, you have chromosomes that will code for the same genes. Those are called homologous chromosomes. So if our gene for spore color was right there on this chromosome, we would have an equivalent gene for spore color right there on that chromosome. Let's say that this one codes for black and this one codes for tan. Now prior to meiosis, there's a duplication event where we actually duplicate our chromosomes. In that duplication event, then we will have our genes in the same locations again. Sorry, that would be a T prime. Okay, so exact same genes, we'll denote that one as B prime and that one is T prime. This one is black, this one is for tan spores. Now these are homologous chromosomes or homologous pairs of chromosomes. This then is a duplicated chromatid, so this is a sister chromatid to that one, and this is a sister chromatid to that one. All of these then have a centromere binding them together in the middle. Now what we want to look at in this lab is the creation of diversity, right? And so if these separate in meiosis, then all of the genes on this chromosome will be the same and they never change. So this whole chromosome goes into one offspring. This chromosome, and this chromosome. To increase that genetic diversity, then crossing over allows us to switch this with this and recombines these genes so that these genes here would be with these genes. So let's say that crossing over occurs right there. If crossing over occurred right there, then this little segment would switch with that segment. This T would now be over here and this B would be over there, and with all of these genes we would have tan spores associated with that. With all of these genes we would have black spores associated with that. Now let's say crossing over occurred there instead. If crossing over occurred here, our B and our T would not switch. Only that and that would switch. So in order for our B and our T to switch, then crossing over has to occur between the centromere and the gene. Let's say crossing over occurs down here, same situation. These two switch, our B and our T do not switch. We don't have recombined genes in that situation. Okay, now let's kind of look at this in a different way. Let's say that these are two homologous chromosomes. Okay, and we're not going to worry about the duplicated partner of each one. These are in prophase 1 of meiosis, so we're intertangling and crossing over is going to occur. So here's one set of homologous chromosomes, here's another set of homologous chromosomes. Now, crossing over can occur then between these two, right? Switching that segment and that segment. Crossing over can also occur on these two, switching that segment and that segment. So now if crossing over is random, we'll just kind of randomly shoot some crossing over events through here. I'll randomly shoot some crossing over events through here. Okay. If you look at these crossing over events, we can see that this scenario is different than this scenario, where the gene is close to the centromere versus the gene being far from the centromere. Over here, if you recall then, that crossing over, if it occurs between the centromere and the gene, then the genes switch. In this case, crossing over only occurred once out of 1, 2, 3, 4 times on this side, but basically once out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 times, right? So very, very rarely do we get crossing over happening between the centromere and the gene, thus causing a recombination to occur. Over here we have 1, 2, 3, 4 out of 10 times that it happens between the centromere and the gene. Okay. When crossing over happens between the centromere and the gene, that's called recombined. And so one that had the switch was called a recombinant. Okay. So if you look at frequencies of recombinants, we would expect a higher frequency of recombinants in this scenario where our gene is farther from our centromere than this scenario where our gene is close to the centromere. Okay, so what we just learned was that when you have a situation where we have two different potential scenarios where the centromere is close to the gene versus the centromere being far away from the gene, in this scenario we would have fewer recombinants seen than in this scenario over here. So now let's say that we want to even quantify that more. Okay, so what we could do is we could say that since every chromosome is a different length, that this chromosome is 100 units long, which are going to be called map units. So this is 100 units long, this is 100 units long. If we wanted to get an exact map unit, in this case, let's say that the centromere was exactly midway in this chromosome, which it doesn't have to be. If that was the case, then if this was at the tip and this was directly in the center, we would have 50 map units from this centromere to this gene. So over here, then, we have fewer than 50 map units. If this is all 50 map units, this might be somewhere around 15 map units. To quantify that directly, assuming that crossing over is random in between those points, what we could do is that we could look at the number of recombinants, okay, divide that by the total number of recombinants plus non-recombinants times 100 to get percent recombination. And percent recombination then would basically tell us how far these are from the centromere. For example, if our percent recombination was 50%, right, we had 50% recombined, that would be exactly this scenario, and our map unit distance would be 50 from the centromere to the gene.